Okay, welcome to our physiology lesson on pregnancy. These are the topics that we're going to cover. We'll start with fertilization, naturally, of the ovum, and growth within the oviduct and transport to the uterus. So the ovum is going to come down the oviduct to the uterus. It's going to implant, and then we'll talk a little bit about maternal recognition of pregnancy and physiologically how the body knows that it's pregnant and what it does in response to that. We'll talk some about the placenta and how that works and forms. We'll talk about some of the hormones of pregnancy as sort of an introduction to what happens after that. And then we'll talk about how those hormones induce parturition, and then we'll talk about the process of lactation. Okay? So that's where we're headed. Let's start with fertilization. So here is the magical organ that has been responsible for so much happiness throughout the world. This is our female reproductive tract. You will notice that during the process of reproduction, as we've described it up until now mostly through the female reproductive endocrinology lesson, we have hundreds of millions of sperm that are deposited in the vagina. But notice that only a small fraction of that number, about 100,000, actually make it into the uterus. And only maybe a maximum of about 50 make it up into the oviduct to the point where they actually have a shot at the prize, and where they can actually fertilize an oocyte. And this process is facilitated by muscular contraction and peristalsis. There's ciliary movement involved and mucus flow, which we've kind of described a little bit, again, in the female reproductive lesson. So the process of fertilization pretty much goes something like this. You have your sperm. It weaves its way through the corona radiata, and it makes it to the zona pellucida. Enzymes are released from the acrosome, which eat away at the zona pellucida, and you form a hole through which the sperm moves its way through until you finally get to the oocyte membrane, where the nuclear material in the head of the sperm is released into the oocyte, and genetic recombination takes place. Okay, just like that. And of course, as this takes place, the second meiotic division finishes, and your second polar body forms. And again, you get the genetic recombination. Only one sperm, in theory, penetrates. And there's a reason for that. We'll describe it. This wall actually hardens. And so you end up with one set of male chromosomes and one set of female chromosomes left after the formation of the polar body. Genetic recombination takes place. So here's a little more detail about how it happens. We have our sperm cell weaving through the residual follicular cells surrounding that oocyte from the corona radiata, and as we approach the oocyte, a rise in calcium inside the sperm triggers the acrosomal reaction. That is, basically what happens is the cell membrane of the acrosome fuses with the sperm membrane, and the hydrolytic enzymes are released effectively through what is one massive exocytosis reaction. And those acrosomal enzymes are released, and they digest away the zona pellucida locally, so that you get this hole that forms. And at the same time, the sperm is hyperactive. Of course, you know that, hypermodal, and it's trying to push its way into the ovum rather actively at that point. The two membranes fuse. The cell membrane of the sperm head with the oocyte membrane, and effectively, again, what you get is exocytosis of the nuclear contents or the chromosomes in the sperm head into the egg. And they enter into the egg cytosol somewhat as you see there in yellow. Okay. At the same time, when this happens, there's a rise in calcium inside the oocyte, and what you have are these granules that are just underneath the zona pellucida. And what happens is those granules, there's an exocytosis that takes place of those granules through the cell membrane, and your zona pellucida hardens in response to that, so that no other sperm can get through the zona pellucida. You end up, in theory, one sperm depositing its contents, and then no other sperm can get through. Okay. That is a cortical reaction, is what that's called. At the same time, that rise in calcium causes completion of the second meiotic division in the oocyte, and you get formation of the second polar body. And genetic recombination takes place with the male nuclear material and the female nuclear material coming together, the chromosomes combining to make the beginnings of a new individual. Okay, so let's talk a little bit about how that results in growth in the oviduct and transport to the uterus. And I would expect embryology will cover this perhaps in a bit more depth, but generally speaking, here's our oocyte here, and we have the ovary here, and the oocyte is expelled through ovulation into the oviduct at day 0. Fertilization, if it's going to take place, takes place just a little ways down the oviduct here, and we just described that. And then the cell begins to divide, and we have a 2-cell stage, and by the time we get to the 4 to 8 cell stage, a moruli has formed, and at the same time you'll notice the oocyte, the fertilized oocyte, has traveled further down the ooduct. Okay? And then finally, we have the formation of our blastocyst, which is about 32 to 64 cells, somewhere around day 5 to 6, and we've hit the uterus. Okay? And this transport, particularly in the uterus, is dependent upon, or the lower part of the oviduct in the uterus, dependent upon cilia, which are sort of brushing the oocyte down the duct. Okay, let's talk very briefly about the process of implantation. First of all, progesterone is required for implantation to take place. You know, it's fairly high at this stage of the female cycle, simply due to the fact that your corpus luteum is around and secreting it along with estrogen. And it probably is necessary to prepare the uterine wall for implantation. The blastocyst attaches to the uterine wall, and what happens is the uterine wall actually reacts to that attachment. And it starts to basically form the female component of the placenta. As we'll see in a moment, the placenta is actually made up of a meshing of 2 different tissues from the embryo on one side and a mother on the other. And once the blastocyst attaches to the wall, the maternal component of that starts to develop, as well as the fetal component. Histidine and prostaglandin apparently are required for that process. And again, the structures of the placenta that forms as a result of this implantation either come from the mother or the fetus. They're they sort of come together and interdigitate, as we'll see in a moment. How does maternal recognition of pregnancy take place? What we mean by that is the female reproductive system has to have a way to recognize itself, the fact that it's pregnant. And it has to do that basically, mostly, because progesterone has to stay high throughout pregnancy. And in order for that to happen, the corpus luteum has to stick around. So something has to tell the mother, keep that corpus luteum there, keep it secreting progesterone and estrogen so that pregnancy can take place and continue to develop. And that's what we mean by recognition. And the bottom line is this is accomplished through secretion of a hormone called human chorionic gonadotropin, or HCG, hence known as HCG from here on out. This is secreted by the embryo. So HCG, again, secreted by the embryo and the syncytiotrophoblastic cells of the placenta. So these are cells of an epithelial covering that surround the embryonic part of the placenta as it develops. So it's part of the embryo. And they are partially responsible for secreting this HCG. And what it does is it prevents disintegration of the corpus luteum so that progesterone sticks around and remains high rather than falling off and instead of the progesterone falling off and you get bleeding, menses, what you get is maintenance of that endothelial, myoepithelial layer, and myoendothelial layer, and you get continued development of pregnancy. What's going to happen is progesterone is eventually going to start coming from the placenta. And when the placenta has basically taken over the secretion of progesterone, the HCG is no longer needed, and it will stop being secreted. Okay, and the corpus luteum is going to disintegrate. So this is how that happens. Here we have ovulation at about day 14. Progesterone is going up because our corpus luteum is forming, and because it's secreting more and more progesterone. And should fertilization and implantation take place, HCG begins to be secreted from the embryo and from the embryonic part of the placenta, and it goes up. It's this green line, and it goes up over time. And because it goes up, this corpus luteum, which would ordinarily disintegrate at the end of the female cycle, sticks around and keeps secreting progesterone, and progesterone remains high. Okay, and HCG manages to do that because essentially, structurally, it's a lot like LH, and it's so much like LH that it binds to LH receptors. And so what happens is the HCG binds to the LH receptors on the corpus luteum, and the plasma progesterone and estrogen remain high because the corpus luteum continues to secrete it, as you can see here. So eventually, progesterone starts to come from the placenta, and it takes over in terms of maintaining this level of progesterone. And as that happens, the HCG is no longer needed, and the concentration falls, and the corpus luteum involutes, and it self-destructs. Okay, the HCG toward the end of this in later weeks of pregnancy is relatively low, but the progesterone remains extremely high, and in fact it has increased through pregnancy, as does estrogen, because the placenta secretes in more and more of it over time, which is necessary for the continued development of the continued progress of pregnancy. Okay, so let's talk a little bit about the placenta. The syncytiotrophoblastic membrane that I talked about, these extensions differentiate into what's known as the chorionic villus. And what that means is they punch through into the maternal part of this interface. So what you have here is your chorionic villus, and it's punched through into this space, the intervillus space. And this actually contains maternal blood. Okay, so what you have is you have this chorionic villus sticking into this space. It's surrounded by maternal tissue, and then that is surrounded by this big space with blood in it. And this is where this once it's established, within 3 weeks of fertilization, this is what's responsible for nutrient and waste exchange between the fetus and the mother. Okay. So what you have here is you have this placenta which has developed, and you'll notice it has a maternal portion, the decidua basalis, okay, and it has the chorion, or the fetal portion. And the interdigitation of these two portions forms the interface between the maternal blood in this intervillous space and the fetal circulation, okay? And materials have to cross this interface in order to get from the mother to the fetus and from the fetus to the mother, okay? So you should note that the presence of the placenta has significant consequences for the maternal circulation. First of all, you have to increase the cardiac output to supply the extra tissue. And second of all, the velocity of flow in this area has to be really low, because in order to facilitate exchange, you want to allow plenty of time for exchange of material to take place here. And because if you have a rapid influx of blood, let's say you have pulses of blood even, you might dislodge the fetus. And we definitely don't want to do that at any stage until delivery is ready to take place. So I mentioned we have this exchange across a couple of layers of tissue. So we're not talking about a little thin capillary layer here. So although some diffusion takes place of small organic molecules like urea, and of course water and electrolytes and oxygen, this can take place across this interface via diffusion. The diffusion would be far too slow. If we tried to get anything bigger than that across this interface, and so what we need are transporters. So we have special transporters for larger carbohydrates, lipids, amino acids, and vitamins, and things like that. Some of these hormones, antibodies, these things don't just diffuse across the placenta. They have to have a pathway to get across. You need special transporters in order to do that, okay? This is how nutrients are carried in and out across this barrier that controls the exchange between the mother and the fetus, okay? You should also recognize that there's a lot of cooperation that takes place in terms of allowing the placenta to secrete hormones. It is an endocrine organ. We have a lot of exchange of material between the mother and the fetus. We have cholesterol and androgens feeding in from the mother to the placenta. We have androgens feeding in from the fetus to the placenta, and these are precursors for these hormones. So the placenta is going to use these hormones to make and secrete progesterone and estradiol primarily, and of course these increase gradually over time over the course of pregnancy, and again that's fairly important in order for normal development of pregnancy to take place.